Today I'm going to talk about preclinical work, but I think it's exciting and we're hopeful will have important clinical implications. Next slide, please. Oh, sorry, do I advance the slide?
You do it.
There we go. This is my conflict slide. So Lynch syndrome is a genetic disease that confers approximately 70 to 80% lifetime risk of developing colorectal cancer. Shown on the left is a picture of Aldrin Worthen, who discovered the disease in the early 20th century, and on the right side is Henry Lynch, who rediscovered the disease actually in the mid-20th century. Shown at the bottom is the first Lynch syndrome family or pedigree, which was originally identified and published by Worthen, actually. And I've seen members of this family, and actually they live in California, in the Midwest, and in Germany. So as the cost of genetic testing has significantly decreased to now where genetic testing is really a couple $100 cost, and more people are sequenced, we now find that Lynch syndrome, which we thought originally was kind of an orphan disease, in fact is much more common. So current estimates from the United States and Scandinavia are that approximately 1 of every 280 or 250 people are affected with Lynch syndrome. In the United States, this amounts to approximately 1.1 million Americans. The genes that, and once again, we're talking here about mutations that go through families. We're not talking about somatic mutations in tumors like we've heard a little about, like the FLT3 mutations in the previous talk. So Lynch syndrome mutations that occur are in DNA mismatch repair genes. So essentially these repair different types of small lesions in DNA, in particular base substitutions, and most importantly for this talk, small insertion and deletion. So these smaller indels, as they're called, mutations, are particularly important because they cause frame shifts. So DNA is a triplet. 3 nucleotides of DNA encode for a given amino acid. And so if you have, for example, a 1 base pair deletion, you cause a frame shift. So this is shown here schematically that if you have a point mutation, this can affect 1 amino acid, which can be, this is meant to be, a schematic, not literal, but a T cell can essentially respond to a particular mutation. However, if you have a frame shift mutation, such as we see in Lynch syndrome, you have many amino acids. These can be actually as long as 80 amino acids long, and consequently you can have sort of robust immune responses. And basically, the idea that you have sort of many of these frame shift mutations is why for DNA mismatch repair deficient cancers more broadly, PD-1 and PD-L1 inhibitor immunotherapy is effective. So a key point is that the mutation rates are so high, and they're not random. They're in specific types of sequences, for example, repeats of adenines, for example, is shown here. So the example I like to use is the TGF-beta type 2 receptor, which is a gene that has an 11 adenine repeat shown on the bottom. And in 60 to 80% of colorectal cancers that have mismatch repair deficiency, including Lynch syndrome, approximately there's the same mutation occurs and the precise frame shift protein occurs repeatedly. So this is not so-called private mutation, but it's something that occurs across many tumors. And because there are actually a number of these types of mutations that are shared, it lends itself to the concept of a vaccine approach. So this is kind of a busy slide, but essentially this describes in mice to understand or to test preclinically the concept of a cancer prevention vaccine. Immunoprevention is exciting, but once again, you have to make sure that this is safe and is effective. So summarizing this slide, we screened with colleagues at the DKFZ in Germany and identified in the mouse recurrent mutations like are seen in people. And we initiated, then tested to make sure that these mutations were immunogenic. So we screened something like 53 genes and ended up with 4 that were recurrent mutations and which collectively tumors in the mouse model, essentially 1 or more of these mutations is expressed in almost all the tumors. And then shown sort of on the right, middle, and right are just sort of a demonstration that in fact T cells do in fact respond to these mutations as would be sort of predicted. Aspirin is used for prevention of colorectal cancers and Lynch syndrome. Aspirin is a non-steroidal anti-inflammatory agent. There was previous studies from Ohio State that suggested that naproxen is kind of like a super aspirin. So it's important in science, obviously, to have reproducibility. So we essentially replicated data from this other group and showed, in fact, that we compared head-to-head naproxen and aspirin. And indeed, our data support that naproxen has better efficacy at preventing colorectal cancers, at least in mouse models. And in fact, we also have, there's a phase 1b trial of naproxen alone in Lynch Syndrome patients that will, I think, be reported at a future meeting, a study that's sponsored by the National Cancer Institute. Consistent with the idea that the preclinical model, the mice are living longer, the tumors are reduced, so the overall number of tumors and particularly the size of tumors actually is reduced in mice that are given naproxen compared to aspirin or the control. So basically, to kind of summarize, we then tested next whether naproxen and a vaccine approach that uses these recurrent neoantigens would prolong survival. And in fact, indeed, we see that the best survival appears to be the combination of naproxen and an immunoprevention vaccine using these frameshift proteins. Also consistent with the idea that these are actually, that these are affecting the immune system. If we look at the T cells that inhabit the GI tract that are important and recognized, we think the earliest cells that transform into cancer, we in fact see that there's an increased response from naproxen. So in conclusion, from this preclinical study, Lynch syndrome is a much more common disease that than we understood previously that confers approximately 70–80% lifetime risk of colorectal cancers, as well as actually other types of cancers, including endometrial cancer, ovarian cancer, gastric cancer, and that this is an important model system in thinking for an immunoprevention vaccine. And basically, if you're ever going to get, the idea of a cancer vaccine, of course, is very attractive. If you're ever going to get this to work, probably Lynch syndrome is actually a paradigm system to study this. That the mutation rates in particular have very immunogenic mutations that are so common, they're shared among multiple tumors and lead themselves to a vaccine type of approach. From our studies suggest that something that's very simple and then hopefully therefore translatable is that if you take as few as 4 of these mutations and vaccinate mouse models with this, you actually see a significant effect. So therefore, the simplicity of this approach means that we think it can be taken forward. And then naproxen appears to be a super aspirin in increasing efficacy in mouse models of Lynch syndrome, and that clinical trials for Lynch syndrome immunoprevention are feasible and will hopefully start in the near future. Thank you.